part 2 of GI function. So what I'd like to do now is march through the processing of a meal so we can see what each segment does in what order and what the stimuli are. And we'll go from there. All right, I won't be able to fit a picture of the entire GI tract on this board, so I'm going to have to do it in pieces. So I'll start with some simple things. Let's start with the upper GI. So I'm drawing just the GI tract, essentially, the GI tract associated structures. Right? Teeth, right? So we're going to bring in our food. We're going to chew it, also known as mastication. Really the only purpose behind that, I mean, some animals do not masticate. Snakes swallow their food whole. The reason to do it, though, is to increase the surface area. For higher metabolism animals, it makes sense to try and extract food more rapidly, or extract nutrients more rapidly from the food we eat. And so increasing its surface area allows the digestive secretions that are going to come in later to act on a larger surface area more quickly. The other secretion, or the other process associated with the oral cavity, is a secretion known as saliva. And saliva comes from a series of salivary glands indicated by these ducted glandular structures. And in humans, this isn't a big deal, but in humans they're broken up into the parotid, the submandibular, and the sublingual salivary glands. The secretions are very similar, not exactly the same, but pretty similar. Label those. And those secretions include mucins, which are, it's a product, essentially acts as a lubricant to the food. Again, so we want to break it up and lubricate it so it doesn't get stuck on the way toward the stomach. Right, it's got to pass through the pharynx and esophagus. So anyway, so we chew the food, we lubricate the food. We do actually add enzymes to it at this point. The salivary gland secretions include a carbohydrase and a lipase. So we have to talk about the four primary nutrients and the enzymes that degrade them. So the four primary nutrients are carbohydrates, proteins, lipids, and nucleic acids. The last one is a very minor component, but it is a component of dietary intake because all animals and plants contain DNA and RNA. And so we need enzymes to specifically degrade each one of those, right? Because enzymes are bond specific. And so we need enzymes specific to peptide bonds versus the ones that hold monosaccharides together within a polysaccharide, right? And so we have a group of enzymes with fairly literal names here. Carbohydrases, proteases, lipases, and nucleases. All of these are secreted as active enzymes, except for the proteases. They are secreted as zymogens. A zymogen is remember we talked about hormones. Some hormones are secreted as prohormones. Well, these are proenzymes. A zymogen is a proenzyme. And the rationale for doing that is that proteases have the potential to degrade the cells that are producing them, right? Chewing up the protein components of the cells that are actually making those molecules. And so they're made as a precursor molecule that typically becomes activated once within the gut contents. We'll talk about a few of those. Okay, so the salivary glands include amylase, which is a carbohydrase. right? So we've got the ability to digest carbohydrates coming with saliva. And there's also one of them specifically produces, one of the glands produces a lipase. Now, having said that, these enzymes are not active in the oral cavity. Unless you have a habit of, carrying food around in your mouth for prolonged periods and not swallowing it, it's just not going to be there long enough. And so those enzymes are added, but then are generally active in the stomach for a more prolonged period. Okay, so we masticate, we swallow. Peristalsis actually starts at the upper esophagus. So we had the oral cavity, the pharynx is basically your throat. And then right below the pharynx, so I'll start it about here, is the esophagus. And what we have, and there are several of these all along the GI tract, are sphincters, right? They're essentially like rings of muscle that act as valves to help control the passage of secretions and food from one location to another. And so we have an upper esophageal sphincter, and then we have a lower esophageal sphincter. Space in there. As you can see, it passes through. Right, this is the esophagus. As I was saying, the peristalsis does start at the upper esophagus. So, and the muscles in the wall there are actually under both conscious control, and there's another set of muscles that are under unconscious control, the smooth muscles. And so swallowing is conscious, but then peristalsis in the same region, the upper esophagus, takes over to make sure that food travels in the, in the direction it needs to. And these are powerful contractions. You can swallow while standing on your head. You get it past that upper esophageal sphincter, and the peristaltic contractions will push that food toward the lower esophageal sphincter. Okay. The lower again, so the esophagus is just a passage, a conduit. It's not really serving a digestive function, other than the contractions that convey it to the stomach. Right, so the stomach down here. Right, so the lower esophageal sphincter is there so that, not so much to keep things from going in here, although some people, there's a condition where that guy doesn't open properly, and food sort of backs up here. But its main purpose is to keep stomach contents from going north during processing, right? And so when we swallow, we want this guy to relax, and the contents, the food that we're eating, to be allowed to enter the stomach. Then we want it to constrict, especially when in between meals. It constricts especially when we're laying down, again, to keep stomach contents from sort of gravitationally take this and rotate it 90 degrees. We don't want the contents moving backwards up into the esophagus. As you're probably well aware, the stomach produces a very acidic environment, which would be dangerous, and it is adapted to defend against that and utilize that. The esophagus is not, and so we need this guy intact. Otherwise, you're going to end up with gastric reflux disease and lower esophageal ulcers, none of which are good. Okay. All right, so now what I'm going to do is I'm going to get rid of this picture, and I'm going to go to a picture that starts here. So let me start with this. Let's have here's the end of our esophagus. I'll add that sphincter in a second. So again, stomach. Here's our lower esophageal sphincter. The opening, if you've heard of it before, the opening is called the cardiac orifice. And this region is called the cardia. This is the fundus. This is the body. This is the pylorus. This is regional stuff that you may have learned. And again, if you had AMP. If not, it's not a big deal. I'm just pointing it out. So now we have our food entering. I keep making it green for whatever reason. Right? Our food entering the stomach, right? Through the lower esophageal sphincter. Now, before this even occurs, let's back up for a second. Before this even occurs, we do have neural control of digestion, right, through the enteric nervous system and the autonomic nervous system and the linkages between them. What that means is that we can initiate GI function before food hits the stomach, right? So that is called the cephalic phase of gastric activity, okay? Gastric refers to stomach, and so your cephalum is your head, so this is basically brain control of stomach activity before you've actually eaten food. So what that means is that it's your body preparing for the eating of food. So in other words, when you smell food, when you're tasting food but you haven't swallowed it yet, even when you're thinking about food, we can initiate the cephalic phase of gastric activity, which is going to be tied to the things I'm about to tell you here, right? This increase in activity of the stomach actions. If you don't actually eat food, but you get that initiation of the cephalic activity, you can get like acid indigestion because you get the stomach sort of doing its thing, but then you never actually add the food, right? And so you get sort of stomach sitting there with all these secretions that it doesn't have anything to do with. And it takes a little while for that to dissipate. Before I get, let me put a couple more anatomical labels on here. We do, remember, this is the pylorus, that sort of exit end of the stomach. And we have another sphincter here, right? And that is the pyloric sphincter. And it controls outflow of the stomach. And the reason to have that is that the stomach is much larger in volume than the next segment, immediate segment of the GI tract, which is the duodenum of the small intestine. So the small intestine comes next. And the first 1.5 feet of it, and the small intestine is very long, more than 10 feet long, but the first 1.5 feet of it is called the duodenum. And we don't want to release, and the duodenum is essential to digestion. We were in the initial phases of activity here. It's all about digestion. It's not about absorption yet because we're not ready yet. We have to like break up the food nutrients into their composite parts. You typically call it a monomer before we can absorb them. So anyway, so we need to do that first. Well, it's really nothing about absorption yet. But we can't release all of this large stomach contents, more than 1 liter in many cases, into a very small caliber and small volume duodenum all at once. So we need a pyloric valve to sort of like release slowly. It'll relax and contract, relax and contract over time to sort of release the stomach contents slowly over a period of several hours if it's a solid meal. If it's a liquid meal, it'll pass through much more quickly. We don't need to delay it quite as long in the stomach, not nearly as long in the stomach. Okay, pyloric valve. We have, and again, this will be the duodenum, we'll follow up on that in a little bit. In the stomach, so when food enters here, we're going to initiate what's called the gastric phase of gastric activity. And it is going to be the exact same thing, exact same actions as the cephalic phase. It's just the stimulus is different. During the cephalic phase, it was the smell, the taste, the sight, the thought of food. Whereas the gastric phase is initiated actually by the presence of food, right? And so now we've got sensory neurons in the wall here that sense stretch, distension, that sense particular nutrients, that sense the pH, and all of those can be tied to activating the digestive processes here. Now those same processes can be activated through the nervous system without the presence of food, right? That was the cephalic phase. Okay, so cephalic and gastric phases are very simple because it's the same things, except the stimulus is different. Okay? Now, what are we stimulating? Okay, so now we have to think a little bit about the wall here, because the wall is not, if we were to take a microscope and zoom in on this wall, it doesn't actually look like this. It's not a straight line. And so I'll sort of blow up one little spot here. We talked about the villi, right? You have villi, but you also have indentations called crypts. Sometimes they're called glands. And so the surface, if I blew it up, it actually looks more like this. Right, where there are projections into, those would be the villi, into the lumen, and then there are pockets. Again, we have to sort of expand the thickness of our wall here, right? So we can, and again, I'm just exaggerating it in this one spot so we can see it, right? This is not a tumor on the side of the stomach here, right? But so we've got these projections into the villi, and then the pockets that are continuous with the lumen as well, called the crypts or glands. And so those are called, in this scenario, they're called gastric glands. The gastric glands and the crypts in general contain all kinds of different cells. These are the cells, they're replacing the cells, they're making new cells to line the stomach or the intestines, right? They're producing antimicrobial peptides. There's a specific cell called the Pineth cell that does that. All kinds of craziness going on. But there are also hormone secreting cells, the enteroendocrine cells. And one of them is called, I'm just going to put a couple different colors down here so to remind us, like we color coded them in the first figure, right? Or in the first video, right? Just to point out that we have different cell types down here. And some of them are endocrine cell types. And I'm going to have to squeeze this in somewhere. I think I'm going to squeeze it in down here, just because I have more room. Right? We have G cells, D cells, chief cells. These are just sort of the primary ones. And parietal cells. Okay? Well, the G cells are the gastrin producing cells. Hormone number 1. Right? And I'm not going to be able to link everything on, write everything on the board here. Just not enough room. So, what can stimulate gastrin? Well, remember, gastrin can go up during the cephalic phase, which means that neural input can stimulate gastrin, but it also can go up during during the gastric phase. Cephalic phase due to neural input. Gastric phase due to food input, meaning that distension or the presence of, typically it's amino acids or peptides, will stimulate gastrin. What does gastrin do? Well, gastrin stimulates chief cells. Rearrange my arrows here. I'm going to go to the other side. So I have to list what they do. So the G cells are stimulatory to the chief cells and the parietal cells. So I'm going to put green plus sign, green for go. Right? Stimulatory to those two cell types. Now gastrin, that's not all gastrin does. Gastrin tends to indirectly stimulate motility. It stimulates contraction of the pyloric valve, right? Sort of saying like, okay, food just showed up here, let's hold it in the stomach for a while. It stimulates GI growth, so reproduction of the lining of the gut, right? Because the more we eat, the more we're going to damage those cells, the faster we need them to turn over. And so it's tied to all of those things. In the stomach, its chief function, no pun intended, is tied to the chief cells and the parietal cells. Well, the chief cells produce a zymogen called pepsinogen. And the parietal cells release, largely, acid, H+. So pepsinogen, we get pepsinogen, so this is coming out of here. Right? Remember, the gastrin is going into the bloodstream. It's not coming into the lumen here. But the pepsinogen is and it's actually hydrochloric acid because it's secreted, the hydrogen ion comes with a chlorine ion, so it's hydrochloric acid. And the two together, you're going to get remember what I said about the proteases. They come as zymogens, so they have to be activated. Well, this one's acid activated. And so the pepsinogen in an acidic environment is converted to the active protease pepsin. Okay, so now we're digesting the protein specifically in the stomach through pepsin production. Now, remember, in addition to this action stimulating the pepsinogen, sorry, I'm out of room here. Get my label for stomach out of the way. we end up with a stomach pH of 1.5 to 3. So very acidic. And so we're getting both acidic digestion, just generic acidic breakdown, right? As well as specific enzymatic digestion. Now, don't forget, who came down from the salivary glands up above here? Carbohydrates and lipases, right? So we're actually digesting everything. Well, at least three of the three big nutrients enzymatically. Nucleases are a minor component anyway, right? Because we got lipase from up above, we got carbohydrates from up above, amylase. We got pepsin here, right? And we now have a low pH, all of which is helping to break down. Now these have to be acid insensitive enzymes. They have to be able to function at a very low pH, and that is true. When we get into different segments, we're going to have enzymes that have a different pH sensitivity. Okay, so we got our gastrin stimulating our chief cells and our parietal cells. What are the D cells doing? The D cells, well, first of all, they produce a molecule called somatostatin. You may recall hearing about that. That is GHIH, growth hormone inhibiting hormone. Forgot about that one. Anyway, it's just the same structure. But what it's doing in this case, I'm going to put red negative sign here, is it is not a hormone. It is a Paracrit factor. These cells are found right next door to these cells, and these cells, and these cells. And this cell, its large function is its production of this somatostatin, which acts in a paracrine fashion to inhibit the G cells. So, when in between meals, right, when we don't have the stimulus on gastrin through distension or the autonomic nervous system, the D cells are dominant, right? The D cells are just sitting there all day long saying, don't secrete gastrin, don't secrete gastrin, right? We keep the acid and the pepsinogen production low because we're inhibiting this. When you eat a meal, this secretion doesn't go away. Well, it probably does to a degree, but it largely just gets overwhelmed by the stimulatory input. And then as soon as that stimulus goes away, the D cells are winning again. Now, the D cells are actually stimulated by a pH change, which would be indicative of being between meals, right? In other words, as the pH falls, in other words, as you're processing a meal and the food moves on and you're continuing to secrete acid, the stomach would become even more acidic. That is a stimulus for the D cells to secrete more, to say, whoa, like you're getting dangerous here. We need to shut this system off, right? And so low pH actually stimulates the D cells even more. Okay. All right, we're ready for, I forgot one thing. The parietal cells also produce a molecule called intrinsic factor, which is, vitamin B12 is very acid sensitive. It needs carriers within the GI tract before it can be absorbed. There's one that comes with saliva, R protein that carries it, protects it down in the stomach. That post-stomach, the intrinsic factor gets secreted in the stomach. They sort of swap places, and the intrinsic factor protects the B protein, the vitamin B, until it gets absorbed and separated from this intrinsic factor. Believe it or not, it is the only absolutely necessary function of the stomach. You can get by without any of this other stuff. Not to say you have normal digestion without any of this. But you absolutely have to have B12 absorption. B12 is essential for red blood cell production, is essential for myelin production, has a lot of really important roles. The good news is the liver contains, on average, 3, 4, 5 years worth of stored vitamin B12. And so even if you're deficient in that hormone or there's a problem with intrinsic factor, you don't actually become aware of the deficiency for quite a long time. Okay, let's get to the duodenum. All right, so I'm going to switch this. Now we have to keep this pyloric sphincter in the picture because we're going to have some actions that play back on that. So we'll have the stomach ending here. Shift it a little bit. No, that's probably about right. And really, I'm not going to go very far here. I'm just going to go to the end of the duodenum. Remember my pyloric valve is here, or pyloric sphincter. I mean, that's still stomach up there. Duodenum. Now what I've got is I've got a duct that goes off this way. And I've got another duct that goes this way. The blue duct is associated with that's the pancreatic duct, is associated with the pancreas. So this sort of leaf-like structure is the pancreas. This is the common bile duct. The common bile duct goes up into the liver. Well, it goes up into the gallbladder, but it also goes up into the liver because you get these ducts that kind of go up and branch, and branch, and branch, and branch into the liver. All right, so this is the gallbladder embedded in the underside of the liver. So draw this sort of big red blob surrounding all this. Right, liver, gallbladder sitting up on the underside of the liver. Liver does 1,000 things. Involved in metabolism, involved in toxin detoxification of things, involved in recycling red blood cells and the iron associated with the red blood cells, with the hemoglobin, nutrient storage. Our key function here, though, is largely the production of bile. Right? So the liver produces bile, which is stored in the gallbladder. The gallbladder doesn't do much of anything. It doesn't really modify the bile. It does, the duct of the gallbladder does produce some buffer, some bicarbonate. We'll come back to that. But it's going to connect. I've got it dashed here because it goes behind the duodenum, behind the stomach/duodenum, to connect here, right? And so I've sort of exaggerated the spacing a little bit, but hopefully you get the idea. So now, it's not called food anymore. It's called chyme, the mixture of food and mucus and enzymes and acid that comes out of the stomach would enter the duodenum. Well, the largest particulate matter that passes through there is about 2 millimeters in diameter, right? So everything's been broken down into there's no larger, pieces of food anymore. It's all been broken down. Again, broken down to a degree, not absorbable yet. And so, we need to process it further, which means we need more enzymes. Now, the problem is we've got this low pH, let's call it pH 2, coming in here. The pancreas is going to secrete a buffer that brings that pH almost immediately up to like 5 or 6. So we're talking about a 10,000-fold change in hydrogen ion concentration on one side versus the other side of the pyloric sphincter, right? Pretty significant. So here comes the chyme, right? Again, I'll just draw it again as a sort of green blob, right? And let's start with one of our enteroendocrine cells that are found in the wall of the duodenum that's going to become activated. Let's start with an easy one. We'll start with secretin. And so I've got a cell here. Secretin is fairly easy in its actions because the easy thing to remember is called nature's antacid. It stimulates bicarbonate, right? Bicarbonate production. Bicarbonate is our primary extracellular buffer. And so that secretin is acting here to increase buffer production. Secondarily, it acts on the gallbladder duct to increase bicarbonate secretion there. Most people don't talk about that much. It's just less significant. So we're going to assume our pancreas is our major producer of bicarbonate. That's going to enter through this pancreatic duct. There's a little sphincter actually right here, which we'll come back to. I'll just draw those two little blobs. It's too small. Right? It's called the sphincter of Oddi, old school name, or the hepatopancreatic sphincter. Hepato, liver, pancreatic sphincter. So it's controlling the flow of the secretions from the liver, bile in the gallbladder, and the pancreas into the duodenum. We'll come back to that because we have control of that sphincter as well, right? But so the secretin is stimulating the bicarbonate production, which is going to bring our pH. What's its stimulus? Low pH. So pH here equals 5 to 6 plus. Right? And again, depending on what your pH was coming out of the stomach, you're talking about a 10,000 plus fold change in hydrogen ion concentration, buffering lots and lots of protons. Now, the rationale for that is the enzymes secreted by the pancreas. The pancreas secretes both buffer and enzymes. Those enzymes are not acidophile enzymes. They are not acid resistant. Better word. Okay, so we've got our secretions coming out here, right? Our chyme and its low pH stimulates the secretin. We get another enteroendocrine cell activated, which releases a hormone called cholecystokinin. Easy way to keep track of that is we call it CCK. CCK does a lot of things. So CCK is, first of all, what's its stimulus? Its primary stimulus is fat in the duodenum lumen, right? Why fat? Well, fat's because it's going to stimulate contraction of the gallbladder. So CCK stimulating plus sign gallbladder contractions. And so we're going to have, remember we store bile there. Bile is then going to be squirted into the duodenum, and bile is an emulsifier. And an emulsifier, so it's made up of what are called bile salts. And what an emulsifier does is it makes things that are normally immiscible, miscible. What that means is like oil and water don't mix. Well, they are immiscible. If you emulsify the fats, then they will stay suspended in water as droplets, right? You've made them mixable, essentially, right? And so the bile salts in bile are amphipathic molecules. Amphipathic, that means the molecule has both a hydrophilic and a hydrophobic end. Well, easy answer here. Bile salts are comprised of a cholesterol end, lipophilic, hydrophobic, and an amino acid end, hydrophilic. And so what happens is they glom onto the fat molecules, fat droplets that are coming with this food, right? So the cholesterol end is sort of stuck to the fat because it's hydrophobic. And the amino acids are on the outside, making it soluble in the aqueous solution in the lumen there. Right? And so we end up emulsifying the fats. We end up with what are called micelles. Micelles are emulsified fat droplets. Bile salt emulsified fat droplets. Why do we want to do that? Well, we can't digest giant blobs of fat, right? The fat would tend to sort of keep coalescing into bigger and bigger blobs. You can't get your enzymes into the center of that big blob of fat to break down those fats for absorption. We need really small fat droplets. Now the enzymes can act on those little, right, smaller surface area to volume ratio in small droplets, allows the lipases, right, which we're going to talk about those in a second, lipases to digest those fats to allow them to be absorbed across the wall of the gut. Again, that's not happening yet. That's going to happen after all of this. So, cholecystokinin stimulates gallbladder contraction, releases the amphipathic bile salts, which emulsify the fats, stabilizing the small fat droplets known as micelles. Okay? And again, micelles are in the lumen. We're going to have other emulsified fats later on. After they're absorbed, they're no longer called micelles. Okay? We'll come back to that. Okay, we're not done with cholecystokinin though, because cholecystokinin has a multitude of actions. So I'm going to put CCK on here. Now, CCK is going to be acting on the pyloric valve. Okay, it's in response to food being present in the duodenum. So we would want to open it or contract it. Contract it. We want to slow down the release of the stomach contents, right? As soon as food shows up in the duodenum's like, okay, hold it. Let's process that. And then a little bit later, it'll open up and they'll be like, hold it, right? So it's going to keep going back and forth between contracted and relaxed, with the CCK tending to contract it and slow the gastric release. CCK actually also inhibits gastrin, which was promoting the opening of that valve, if you recall, right? In the previous picture. Okay, that is actually what's called, or that's a part of what's called the intestinal phase of gastric activity, because now we have intestinal hormones feeding back to control the stomach, inhibiting gastrin, inhibiting gastric release. Right? And so the intestinal phase is a shut off the stomach phase of stomach activity. Right? And all these phases overlap to a degree, or can overlap to a degree, so they sort of blend. But once we are in the intestine with food, we're going to start some of those inhibitory actions through these hormones. Okay, where else does CCK function? CCK acts on the pancreas and stimulates all types of enzyme secretion. Right? So proteases, lipases, carbohydrases, and nucleases, all of them get dumped in here. And again, the protein will be secreted as a zymogen. In this case, it's trypsin, trypsinogen is the precursor, right? Whereas it was pepsinogen in the stomach. It's a different protease, but it's still a protein digesting enzyme. So we get all of our enzymes in here. We're getting our emulsified fats, or our fats emulsified by the bile salts. CCK is also, let's see, what am I missing? CCK 1, 2, 3, I need 4. CCK also acts on does that look like brain? Sort of. Temporal lobe. You get the idea. CCK acts on hypothalamic satiety centers. So your hypothalamus has neurons that are sensitive to blood nutrient levels, blood hormone levels, and those centers are tied to stimulating either satiety or hunger, depending upon what the stimuli are. CCK is one of the potent satiety hormones. It tends to make you feel full. Because of that, because of its stimulus, which was fatty acids in the duodenum, that tends to make fat a stronger stimulant of satiety. You feel full faster on a fatty meal versus another kind of meal, another composition meal. Interesting to note that at least some people with bulimia show lowered secretion of CCK. In other words, they lack a satiety factor. If you're unfamiliar, bulimia is associated with binge eating, where people eat beyond typical amounts. And again, in these cases of hyposecretion of CCK, they're not getting a satiety signal that normal people get, and so they sort of eat and eat without that feeling of fullness. And again, there's a lot of different components that go into that, but in people that are very insensitive to CCK, that can be a cause. Okay, let's see, what do we need? We need, I'm going to add another molecule here that is not, it's not considered a hormone. It's called a Neuroprine. It's more like a nervous system, enteric nervous system neurotransmitter, but I'm going to add it anyway. because I want to mention its function. VIP, vasoactive intestinal peptide. It kind of tells you, right, that was how its actions were first discovered. Vasoactive means it constricts blood vessels. Well, that means it constricts smooth muscle. And so what it's tied to in GI function, which they discovered later, was that it contracts the smooth muscle and helps with segmentation and peristalsis. Right? So VIP is associated with mixing and propagation of food along the GI tract. The last key secretion, again, not a hormone, right? So far we have 3 main hormones, right? Gastrin up above, secretin, cholecystokinin. Here comes number 4. There are only actually 5 hormone, GI hormone, only 5, but 5 key GI hormones. So there's lots of other paracrine and neurocrine factors, but only 5 are considered hormones, i.e. circulated blood levels that would qualify them as hormones. And this one's called GIP. Well, initially it was called GIP because they called it gastric inhibitory peptide. And what it did, or does, and it's to a very minor degree now that we know more about it, right, and I'm putting arrows here. It's hard to, like, if I put a plus sign on here, you have to know what the plus sign means relative to the sphincter. Right, but CCK and GIP both stimulate contraction of it. They slow gastric release, whereas gastrin tended to open it up. Right, but so there's your gastric inhibitory peptide. It inhibits gastrin to a degree as well. Intestinal phase of gastric activity, intestinal secretions feeding back to inhibit the stomach. But a GIP also initiates weak contraction of the pyloric sphincter, again slowing gastric release. What's to keep the acronym working, they switched its name. They found out it does something actually much more important. It's called glucose-dependent insulinotropic peptide. Glucose-dependent insulinotropic. So it's stimulated by carbohydrates, glucose. Insulinotropic means it stimulates insulin secretion. So think about that, and it's a peptide, right? GIP. So think about this. You're going to eat a meal that's high in carbohydrates. Those carbohydrates are about to be absorbed and cause us what's called a postprandial spike or postprandial hyperglycemia, a spike in blood sugar levels. We don't want to have a postprandial hyperglycemia. So to try to avoid that, we sort of proactively stimulate a hormone that stimulates, well, and I could have the arrow go to the pancreas, because remember that's where the insulin secreting cells are. I'll just put it here. Right? And so the GIP stimulates insulin. Insulin stimulates a bunch of things, but its big thing is that it stimulates glucose transporters, skeletal muscle, and fat tissue. And so as these carbohydrates that stimulate the GIP are absorbed, the cells are ready and able to absorb those sugars into the cell to keep the extracellular glucose concentration from rising dramatically. And that's found to be the much more potent action of GIP, not this one. Last one I'm going to throw on here, and this one is actually found pretty much anywhere on the tract, the GI tract, and that's a hormone called Motilin. Motilin is kind of active when all these other guys aren't. So Motilin is sort of the between the meal hormone, and smooth muscle in the GI tract is electrically coupled, meaning it tends to work as a unit, right? There are gap junctions between the cells. And kind of like cardiac muscle, right? It acts as contracts as a unit. It's not quite the same degree of connectivity, but still they're connected. And so we can get these sort of waves of contraction that travel through the GI tract. That happens much faster and stronger during the meal. But in between meals, right, we don't want the whole system to just sort of shut down. That's when Motilin is more active. It's actually inhibited by a meal. But in between meals, the Motilin goes up, and Motilin stimulates these sort of very slow and actually fairly strong wave-like contractions that start at the stomach and travel on the entire length of the GI tract. And think of it as sort of like a housekeeping hormone. It's causing contractions that tend to just sort of like make sure that, you know, there's no GI content. It's just sort of getting hung up somewhere in the tract, right? It's sort of like flushing everything towards the end of the system in a very slow wave-like contraction. It lasts actually about an hour and a half, like one wave of contraction. Much, much slower than contractions, peristalsis during a meal, as a meal is being processed. Okay. Whew. All right, I think I'll stop there and then pick up explaining the end of the absorption and the end of the GI tract, which really doesn't involve a lot of hormones, but just to complete the story, I think I'll put that in a separate video.